Good afternoon, ladies and gentlemen. I request Professor V. Mohan Kumar and Professor Ratna Sharma to please be seated on the dais. Welcome to Dr. Kamla B.K. Anand Oration for 2025. To start the evening, I would like to request our Dean, Professor K.K. Verma, to welcome Professor V. Mohan Kumar. I will now request our Dean, Professor K.K. Verma, to introduce the oration.
Thank you. Honorable Professor B. Mohan Kumar, distinguished members of the faculty, staff, and I see very eminent our ex-faculty members, the deans, the heads of the departments, Dr. Kushum Verma, Madam Nair, and so many other people who have graced the occasion. On behalf of the director who got busy with something urgent, he has to go, but he is coming back shortly. And the All India Institute of Medical Sciences, the family of the AIIMS, it's my profound honor and privilege to welcome you all to this Dr. B.K. Anand Oration, which is called Dr. Kamla B.K. Anand Oration. This oration was instituted by Professor B.K. Anand, who was the ex-professor and head department of physiology, and he was the second dean of the institute from 69 to 74, which means he was probably for the entire tenure of 5 years. He made the donation and instituted this oration in the name of Kamla B.K. Anand Oration. I think the people of my time and the time before me, they have met him and also known Dr. B.K. Anand quite well. I had the privilege of meeting him as a student, though I came much later after his superannuation. But on social occasions, I got a chance to see him, to meet him. He established the Department of Physiology at the Institute and made significant contribution in the field of physiology. He was a person of international repute, a very eminent personality who contributed immensely in the field of physiology and particularly in the neurophysiology. This award is given to a scientist, an eminent person in the field of neurosciences from India, or the people have been chosen from abroad also, who have made, again, the significant contribution either in the field of basic or in the clinical discipline related to neurosciences. In the past, the orations have been delivered by, I think, 17 people before Dr. Mohan Kumar. So he is the 18th one who is doing this honor to us. And the list is very long. I will just read out few names like Professor Ito, Dr. Tandon, Dr. Ramamurthy, and then, of course, the list goes on till it is reaching today to Dr. V. Mohan Kumar. So, sir, it is our honor and privilege to welcome you to this oration. And I invite the head of the Department of Physiology to introduce you, because she would know better than what I can describe you. Thank you.
Respected Dean Sir, Professor Kusum Verma, who was the former head and professor of Department of Pathology and the proud daughter of Professor B.K. Anand, Professor Kumar, our beloved formal HODs who have graced this occasion, Professor Usha Nair, Professor Bijlani, Professor Deepak, Professor Kochar, and our very favorite senior professors, Professor Mahapatra and Dr. H.N. Malik. Welcome to this oration that is being held in the loving memory and loving and proud memory of Mrs. Kamla and Professor B.K. Anand, who laid the foundation of Department of Physiology at AIIMS, New Delhi in 1956, ensuring highest standards of undergraduate and postgraduate teaching and an equally conducive environment for research. I can say without doubt that we are holding on to the standards laid by Professor Anand with passion and enthusiasm. It is my pleasure to introduce the orator of the prestigious Kamla B.K. Anand Oration 2025, Professor V. Mohan Kumar, who is the 18th orator in the series of prominent basic and clinical scientists from India and abroad. Professor Mohan Kumar is our former professor and head of Department of Physiology. And post his retirement from our department in 2006, he took over as emeritus scientist of the CSIR and ICMR at Sri Chitra Tirunal Institute for Medical Sciences and Technology, Trivandrum. He is the recipient of numerous awards that includes 2 Lifetime Achievement Awards and prestigious orations in the field of physiology and neurosciences. Professor Mohan Kumar has worked extensively in sleep research laboratory in the Department of Physiology from 1977 onwards. Professor Mohan Kumar was the first to map out the caudal brainstem EEG synchronizing areas. Further, the role of preoptic basal forebrain in sleep-related functions, reproduction, and thermoregulations were established by him in the lab. Professor Mohan Kumar pioneered in undertaking neural transplantation in experimental animals, as well as fMRI studies in conscious rats showing that medial preoptic area was activated during slow wave sleep. A chamber was designed by Professor Mohan Kumar to study sleep wakefulness, behavioral thermoregulation, and several other physiological parameters telemetrically in unrestrained animals to demonstrate that neural structures implicated in body temperature and sleep regulation are overlapping. Further, at Sri Chitra Institute, Professor Mohan Kumar established Comprehensive Center for Sleep Disorders with a human sleep lab for polysomnographic recordings in 3 patients simultaneously. He initiated translational research based on his findings on animal research at AIIMS Delhi with the role of environmental temperature to deal with insomnia as the main theme of the study. He also gives public lectures to increase the general awareness about sleep disorders and has authored more than 190 publications to his credit. With this brief introduction, very respectfully, I welcome Sir Dr. Mohan Kumar to give this prestigious oration to the August gathering. Thank you, sir.
Respected Dean, respected Head of the Department of Physiology, and without naming all those distinguished people who have come here, I pay my respect to them for taking their time to come to this oration of mine. My title of my lecture is Our Journey Through Neural Regulation of Sleep. Our journey is not my journey. It is the journey of my students, my colleagues. I just happen to be along with them. Now, when I was doing my post-graduation in physiology at Madras, now called Chennai, the professor suddenly asked a question: who is the greatest physiologist in this country? We all started looking at each other. He said, it is Dr. B.K. Anand. He is a neurophysiologist and he is working as professor
at All India Institute of Medical Sciences. At that time, I decided where to go. Now, I finished PhD under Dr. Sinha and Dr. V.K. Anand. I was lucky to have got a faculty position. At that time, lecturer was the first faculty position, not the assistant professor as it is today. Then, slowly I realized that I was not on top of the world. Slowly, frustration started building up. I was designated to work in environmental physiology, where my heart was not there. I tried to do my level best, but I couldn't produce anything worthwhile. So, unfortunately, Dr. Anand also took voluntary retirement. 7 years passed where I was reaching the climax of my frustration. I decided to leave Delhi. I had made secret preparations for moving over to Bangalore, though I know I will not get any sleep research position. But I thought something in neurophysiology is better than nothing. Now, then suddenly I got an offer from Professor Mancia of Milan. Professor Mancia is the disciple of Marusi, who is the inventor of ascending reticular activation system. He told me very clearly that this is a very, very meager amount. Think twice before accepting it. I did not have to think twice. I just grabbed it. Now, after reaching Milan, I realized that it was really a very small amount. It was not even enough to pay for my hostel rent. I found a sleeping accommodation in the attic of a multi-storied building where there was no air conditioning. In Milan, the temperature during winter used to go several degrees below zero. And there was not even a bathroom, a common toilet. I used to go to the department every Sunday early morning and have my bath. So bath got restricted to once a week. And food was bread and butter, occasionally a little bit of cheese. Then I got my first publication in brain research. Now I go through, spend a few seconds on describing what this publication is about. It is postsynaptic changes in sensory motor cortical neurons during brainstem reticular activation. Now, this is, as you can see here, this is the brainstem reticular formation. It projects to the cortex through the thalamus, and at that point in time, it was a belief that sleep is a passive process, and decrease in the activity of reticular formation produces sleep. And we were trying to study the role of reticular formation, the anterior portion, which primarily produces desynchronization. And also we were trying to look up at some place in the reticular formation which could produce a sleep. So, as you can see here in this picture, it is the encephalo preparation on which we studied. We had the stimulating electrodes here, and we had the recording electrodes in the cortex. And going forward, we saw that stimulation of rostral brainstem reticular formation produced firing of cortical neurons. These are the intracellular recordings of cortical cells, and we had the thalamic neurons also showed firing. And this was done after coming back to India, and I will just come to it in a few seconds. And We also found that there is on caudal brainstem stimulation, you could get EPSV, IPSV sequence in the cortical neurons, and along with the EPSV, there were firing of this, and this synchronization along with this thalamic neurons were inhibited. Now, responses produced by the caudal brainstem reticular formation was abolished after thalamic lesion, thereby showing all this synchronizing response coming from the brainstem is passing via the thalamus. And these are the publications we had on primarily based on the work done in Milan, and then these are the work done in India primarily by AIIMS. AIIMS was the PhD student who is registered not in the Department of Physiology, but in the Department of Neurology. And I was thankful to Professor Ahuja for letting one of the rooms, they used to have some rooms in the ground floor, and one of the rooms was converted into a neurophysiology lab. And before Dr. Maheshwari joined, a few months, Professor Ahuja was there as the head of the department. I should also thank the ICMR for giving the funds for not only as a stipend or fellowship for AIIMS, but also the funding for establishing the lab in the Department of Neurology. Now, then on my return, two excellent students joined me. They were Sujit Sigda and Neelam Bagga. And with them, I had my first two publications from India. So their joining was also not without a drama, but that's immaterial. On the strength of publications in India and abroad, I got projects from ICMR, CSIR, DST, DBT, and AIIMS, and also got students and equipments for establishing the lab. Now, after that, focus of our research was on the regulation of sleep by hypothalamus, primarily the medial preoptic area. I cannot spend some time on explaining why I took all these things due to lack of time, but I straight go into the responses that we got on further studies. Why hypothalamus at that point in time was considered that by publication of Marusi, he said that sleep-waking cycle is primarily by the hypothalamus. We also found that when recording microelectrodes in Sujit Sigda's work, some of the neurons, he was primarily recording from ventromedial hypothalamus, but some of the electrodes by mistake went into medial preoptic area, and we confirmed after postmortem, and they were showing changes with sleep wakefulness. And similarly, in Neelam Bagga's study, we tried several of these neurotransmitter, adrenergic, serotonergic, cholinergic, GABAergic, glutaminergic, agonists and antagonists, and time does not permit us to give all these transmitters that we have studied, and our focus will be just on this presentation on the noradrenergic input, which will be presented in due course of my presentation. Now, here we first started studying the activity of ventromedial preoptic neurons during desynchronization and synchronization, and you can see there is a decrease in the firing of many of these preoptic neurons during desynchronization, and many of the neurons showed increased firing during synchronization. The caudal and rostral brainstem also produce similar changes on stimulation with the synchronization. There is increased firing in many of the neurons. With desynchronization, there is inhibition of firing. So it could tentatively conclude that medial preoptic area is crucial for sleep maintenance and regulation regulated by caudal and rostral brainstem. And this particularly was a study of B.N. Malik. And then we had to change the strategies, because further studies were mostly conducted on rats. Earlier studies were conducted on encephaloisol cat preparation, which I briefly showed in the very beginning. And the objection was not coming all of them from the animal activists, but also from the funding agencies. Now, so we have to change the mode of research. Instead of traditional methods which were employed, which was traditional methods for electrical lesion and electrical stimulation, we started employing chemical stimulation and lesion. And it has several advantages, which I'll be briefly touching through the intracerebral injection. The intracerebral injection is something is like that. A cannula is implanted on the rat, and the cannula is covered with a stellate. And after the animal has fully recovered from the operative procedure, we introduce a cannula through which various transmitters, its agonists, antagonists, neurotoxins could be of several types, one which particularly destroys the neurons, and other which could particularly destroy the afferent input which is coming to the injected area. And also we improved our technique of recording. That is, we introduced the telemetric system of recording, and telemetric system which became available in the market just at that time. And it was only one manufacturer which was making the telemetric system, and he was very happy to supply that, because he could get a feedback properly from one who is working on his system. And here I should say telemetric system is of extreme importance, because earlier it used to be that we used to place plugs, implant plugs on the head of the animal, and through the plugs we used to take out leads, and through the leads we amplify the signals and record. That means the animals move about with dangling wire, and which is not a good thing for a proper sleep recording. So we had this system, and as I said earlier, first of the transmitters that we started with, and about which we are going to mainly talk about in this presentation, is the norepinephrine system. So this is the system. This is the work of Subimil Datta, and in fact, incidentally, it happened that Subimil Datta was one of the earlier orator of this very same thing. And I forgot to tell you before that, Sujit Sikdar was another person who gave this record, and we are proud of them. Now, noradrenergic antagonist, when it is injected, it produces EEG synchronization. So it good enough evidence to show findings suggest that noradrenergic receptors in the medial preoptic may be involved in regulating sleep. This is the work of Professor Ratna. So we had some unexpected findings with her studies. So earlier I said that norepinephrine, when injected into the medial preoptic area, produce wakefulness. So what we thought was this norepinephrine input or noradrenergic input coming to the forebrain comes through two different fiber bundles. One is called dorsal bundle and other is called the ventral bundle. So we expected that if you have the ventral bundle, noradrenergic bundle lesion, you should get something opposite, or at least you should not get wakefulness. But we got increased wakefulness. So we were taken aback, but we went ahead with the protocol that we had made earlier before the study. And the protocol was first lesion, the ventral noradrenergic bundle which is supplying the noradrenergic input to the preoptic area, medial preoptic area, and then inject norepinephrine into the medial preoptic area. Then we found that it induced sleep. So what is the explanation for it? Now, it could be that is the norepinephrine injected in the medial preoptic area acting on the pre or postsynaptic receptors. It could be possible because it need not necessarily act or work in the same fashion as normally released transmitter as a nerve terminal. A massive input of norepinephrine input is coming, can act at any or both the sites, and what will be more prominent at that point in time is nobody's guess. So at this point in time, I take my opportunity to thank Dr. Gomati Govindad and Professor Shashibhadra from the Department of Anatomy, who gave all their support not only in this work and further works forward. So the question is, what is norepinephrine acting? If it is diagrammatically shown like this, let us assume that norepinephrine released by the noradrenergic fibers produce sleep by acting on the postsynaptic alpha 1. This is just let us hypothesize. If it is the norepinephrine released here, it goes and acts on the alpha 1. The alpha 1 receptors are predominant receptors in the postsynaptic membrane, and alpha 2 receptors are the predominant receptors in the presynaptic ending, and alpha 2 receptors are auto-inhibitory receptors. Now, if that is so, if you again assumption that if you destroy the norepinephrine terminals, and then what we should get, there is no transmitter coming, and we got arousal response. Is it because there is no transmitter coming? That's one way of thinking about it. Now, alpha 2 receptor, adrenergic receptors in the NE fibers, as I just said, are auto-inhibitory. And if this norepinephrine injected is acting on the auto-inhibitory alpha 2 receptors, then you could actually produce a situation that there is at the terminal, there is a decreased release or decreased availability of norepinephrine to act, and that could produce arousal. Now, this hypothesis can be proved only by application of alpha 1 and alpha 2 agonists and antagonists at the middle preoptic area. And this is the work primarily of Ramesh and Vetrivelan. And coming back to the earlier, what we found is norepinephrine producing arousal, and then destruction of it, and after that injection, norepinephrine is producing sleep. Then yohimbine, which blocks the alpha 2 receptors, produced here, as we could see, it produced sleep because it is not allowed to have auto inhibition, and that means an increased release of norepinephrine at the site of action. And application of methoxamine, which is an alpha 1 adrenergic agonist, and that also further confirming that alpha 1 receptors in the middle preoptic area sleep inducing. And the results showed that alpha 1 system is primarily inducing sleep. There are many other receptors. We tried all of them and found that no other receptor is really acting in this role at the middle preoptic system. I should also thank at this point in time Dr. H.N. Malik, not only because he is one of the authors, not only because he is one of the co-worker, he is the one who stood by me even during my difficult periods. Now coming to the Ramesh's work. Now Ramesh, what he did, as I said earlier, that we can also inject through the cannula the neurotoxin. there can be different neurotoxins which can act either on the neurons or the afferent terminals which are coming there. This 5-hydroxydopamine is a neurotoxin which destroys the afferents which is coming to the middle preoptic area, and that produce decrease in sleep, further showing the hypothesis that we are following. The noradrenergic terminal is actually a sleep inducing terminal at the middle preoptic area, and this also produce an increase in the body temperature. As I said earlier, any manipulation that you do, either lesion, stimulation, anything in the hypothalamus or areas around, is an area with so many control systems. You just cannot have anything in isolation. Simultaneously, many things will be happening. One of the main things which is happening is the alteration of the body temperature. And this is one thing which we followed further, and they followed further in such a way that we found that behavioral thermoregulation is an important component which is coming into picture not only for thermoregulation but also for sleep regulation. So for that, we produced a multi-chamber system with 3 different temperatures maintained in 3 different compartments, and there is a flap door here. Through one chamber to the other, the animal could freely move from one end to the other, and all of them are implanted with the telemetric system, so you can simultaneously record the sleep and wakefulness. On the base, there is what is called a vibrator, which also would record the activity of the animal, where the animal is staying, what is the kind of activity it is having at the point where it is staying, and also there is containers for the recording the food and water intake. And also we could also record the temperature of the animal, the brain temperature, the body temperature, and everything by telemetric system. So just before going to it, I can show you what it happens when the animal is given free movement within this chamber. It preferred to stay in 27 degrees centigrade. So that means it is the preferred temperature of the animal which is having free movement between all these terminals. At this point in time, in order to reduce the time of presentation, I just skip several of our work and just make a major statement. Preoptic thermoreceptors are not involved in the temperature selection of the animal, because we know the preoptic area has the thermoreceptors. So it is natural for one to think, could be it is the preoptic thermoreceptors are responsible for all these thermoregulatory system or thermoregulatory changes the animal is making, but the thermoreceptors are present not only in the preoptic area, all over the brain, and especially all over the body also, even in the internal organs. So now after making that statement, I just come back to alpha adrenergic system, which is the alpha-1 adrenergic system about which Vetrivalent has done extensive work. As I have said a few minutes back, injection of methoxamine, which is the alpha-1 agonist, into the middle preoptic area produce sleep. Now, along with this sleep which is introduced, we also found that the animal preference, I mean, I am not showing the 3 curvatures of where they were all in when they were given freedom, but just to reduce or make the projection simplified, I am showing that the thermal preference quickly changed over to that of a warmer environment. So warmer environment thermal preference is also one accompanying the selection of sleep or induction of sleep. So all this time I was just giving one example of one input coming to the middle preoptic area. Primary target was middle preoptic neurons. And what will happen to the all this sleep and other changes if you lesion the medial preoptic neurons? In order to produce lesion selectively of the medial preoptic neurons, we have another important neurotoxin, which is called N-methyl D-aspartate, NMDA, which is in short form called. And when you inject NMDA, it destroys all the neurons there without affecting fibers of passage or the terminals which are coming there. And on injection of medial preoptic area neurons, we again got the change in sleep in such a way that it reduced sleep. So earlier conclusion that the alpha-1 receptors are inducing sleep, okay, it is by acting on the medial preoptic neurons, and the destruction of medial preoptic neurons also produced sleep. Now, it also resulted, like what we have seen in terms of alpha-1 injection, it also produced increase in the body temperature. Now, change in thermal preference ensured the recovery after medial preoptic lesion. That's one other important finding is that rats prefer 30 degrees centigrade after medial preoptic lesion. Now, this was happening along if it is in an animal whose sleep has been already reduced by lesion of preoptic neurons, then it had a slight tendency to increase or restore back the sleep. So, in other words, the neurons or areas or rest of the brain or rest of the body itself is trying to restore sleep. So, in between there is a tendency for all animals and living beings, I cannot say living beings, all animals to restore its sleep by hook or crook. And here it is trying to prefer a warm environment and then increasing its sleep. And body temperature also, earlier we could hypothesize increase in the body temperature which is happening is also a kind of mechanism initiated by the body in order to restore sleep. And also we can always see that whenever we have increase in the body temperature, there is always a tendency to increase sleep. And nighttime increase, this increase is happening as the rat is a nocturnal animal. That means it normally is active at night. And if the sleep has to be increased, the most convenient place to act is when you have increased wakefulness. And bouts of sleep happening at night actually reduce the circadian sleep-wake cycle in these animals. Now, why as we have said in the very beginning, it is not very scientific in order to just look at one of the parameters that you are focusing on, and that may land you up in a kind of a wrong explanation of what your assertion is. There is a whole lot of change which happened. This is a study of Joshi John, and you can see the locomotor activity increased, the body temperature increased, the body weight became low and maintained low throughout the animal. Now, while we were studying the medial preoptic area, there was another group in Harvard, which was they were studying the role of lateral preoptic area. And these people came across a few neurons in the ventrolateral preoptic area, which is CFOS active at the time of the sleep in mice. And they declared to the world that ventrolateral preoptic area is the sleep regulator in human beings, in animals, in all these probable places and probable species. Now, what we have found is we are primarily so far studying the ventromedial preoptic area. And this kind of a assertion by this Harvard group probably because they are a Harvard group, and their voice is always heard better than any saner voice coming from the rest of the world. And then we studied what happens to the lateral preoptic area is destroyed, which includes the ventrolateral preoptic area also. Then that also produced reduction in sleep. Not that their observation is wrong, their interpretation was wrong. What they were trying to do is, just by seeing few CFOS active neurons, they are declaring to the world that that is the sleep regulating center. Later they themselves realized that their observations were wrong, but they were not prepared to admit to the world that they are wrong. But they went ahead by seeing, they found that the entire lateral preoptic area is important for sleep, and even beyond that. Then what they do, the rest of the publication, they said extended ventrolateral preoptic area is important for sleep. There is no term in anatomy called extended ventrolateral preoptic area, but unfortunately the scientific world accepted that because it is coming from, I mean, my blood boils when I read their articles. Anyway, for them, most important thing is self-glorification, and telling the truth is probably one of their last priorities, or if at all there is a priority. So here we can see that same thing as happening in decrease in the sleep is happening after medial preoptic. Increase in the temperature is also happening after medial preoptic. The magnitude may be different, but there are some differences. Medial preoptic area lesion rats preferred higher ambient temperature, whereas we also studied lateral preoptic area and the medial septum, which are areas around the medial preoptic area, and we found that they preferred lower ambient temperature after the lesion. So there is a difference between the two, and thermal preference also for a lower temperature of MS, medial septum and lateral preoptic lesions, and higher temperature of medial preoptic lesion ensured sleep restoration. So this thermal preference of the animal is an important component of thermal regulation, not only a thermal regulation, but also for sleep regulation. Now, thermoregulatory ability of preference of the animal is restored. I mean, it is also because there is a failure in the medial preoptic area lesion animals. When they are exposed to a cold environment, their body temperature drops. That means there is a failure of restoring of temperature when they are exposed to a cold environment. On the other hand, the lateral preoptic area lesion and medial septal also, there is a failure of body temperature regulation when they are exposed to higher temperature. And that's the main difference. And then we use several other techniques. I mean, one technique alone will not give some solid confirmation of what you are hypothesizing. And to that, I must thank Professor P. N. Tandon and Professor Nick Bhargava for the institutional support, because they got an MRI. They probably the first MRI which came to Delhi or probably whole of India, and they had to work very hard to get this MRI. And the beauty of it, they did not just get an MRI for patient study, they also got an MRI for animal research. And with that, we made use of their support and provided by the institute, and we studied later. Earlier we used to, after lesioning of an area, we used to do histology and locate the area of lesion. But it is also possible to see the lesion of the lesioned area by the MRI, and also I should say this is an excellent example of interdepartmental collaboration. And I should particularly thank Professor Raghunathan. And he was a great support to us. He was a gentleman. He's a great scientist. And then later, we had a DBT project about with Rakanath and also as one of the co-investigator, and we want to dedicate this, whatever we achieved in this study, to late Dr. Raghunathan. And this is the stereotaxy. So animals cannot lie quiet in the NMR machine, but you have to have the training of the animal, also a head fixation for which we need MRI compatible stereotaxy. That is one of the technical innovation that we made. And then we also saw fMRI, that the activated areas include the preoptic area, and we also found that many other areas like frontoparietal cortex is also activated with sleep. Now, Dr. Tandon and Dr. Gomati Govinath had a major project on neural transplantation. They were kind enough to include me also into the project, and using their project, we also tried initially a few experiments to establish the functional connectivity of the neural transplant with the host. And I am not presenting that. And I straight away come to the area that we were primarily looking for, that what will happen if preoptic tissue from fetus is transplanted to the preoptic lesion animal. And we see from this curve that the red line showing after transplantation, there is a recovery of sleep. And the body weight showed recovery also after the transplantation. So, before going further, I will just conclude some studies and assessments that we have made, and we come to the conclusion sleep is a global phenomena, on the basis of which I have published a couple of papers. And middle preoptic area is part of the sleep maintenance. So this is, if you, in this picture, here is the middle preoptic area, around which you have the lateral preoptic area and the middle preoptic area. Sleep maintenance is by the middle preoptic area. Sleep initiation could be probably by the lateral preoptic area or areas around the middle preoptic area. Even if you look at the insomnia, I mean, it could be classified in either it's a problem of sleep initiation or sleep maintenance. So you have some anatomical basis of this kind of a difference that you see. Then sleep regulatory and thermoregulatory networks are intimately related, and an important component of sleep regulation is behavioral thermoregulation, which is done not only by the thermoregulatory network but also by the cerebral cortex and elements of limbic system. So the POA regulates not only the sleep and body temperature, but also initiates this function with or integrates this function with several other homeostatic functions. Now, ability or desire to have a normal sleep is inbuilt, and the whole system, whole brain, and even rest of the body works towards attaining a normal or ensuring that the animal or human being has a normal quantum of sleep for better functioning. We also had an international conference in 1992, during which we formed the Indian Society for Sleep Research, Asian Sleep Society, with all those Asian sleep societies, representatives who came onto this area. And it is necessary for us to have these associations to exert ourselves and or show to the world that we are also there and not to be totally neglected. These are the areas where Asian Sleep Research Society had their triennial conferences. We also developed two journals. One ASRS journal is Sleep and Biological Research, which is already got PubMed indexed. And this is Sleep and Vigilance, is the ISSR journal, which is yet to get PubMed indexing. So it was difficult for me to think of a life after retirement from AIIMS. I retired on September 2006. I was not unhappy about it. I have made plans for my future. My plan was to go back to Trivandrum and start agriculture on the ancestral properties that we have inherited. But suddenly, fate had something different in store for me. An accidental meeting with Dr. Radhakrishnan, the professor, HOD of neurology, and the director, Dr. Mohan, changed the course of action, and I came back to Delhi and applied for a emeritus scientist position with the ICMR and CSIR. I got both, but I accepted CSIR one because it has some advantages over the other one. And I continued there as a visiting professor for a few more years after this emeritus scientist position is over. I was also appointed the chair of the scientific advisory committee of Sri Chitra, and I was also lucky enough to be appointed the chair of the Kerala chapter of National Academy of Medical Sciences. And there we planned to have a comprehensive center for sleep at Sri Chitra. So we made a new beginning, and the Sri Chitra is like, it has something like CN Center, which we have neurology, neurosurgery, cardiology are the main specialties. And it also has an Achievement Center for Health Science. It has got an important wing, which is called the Biomedical Technology Wing. And so to say that this is the only medical institute under the DST. And we made a new guest house was made for the Sri Chitra, and there is an abandoned guest house which was there. We converted it into a sleep center to begin with, and started animal research facility and also polysomnographic facility and sleep disorder clinic. Animal research being on the first floor and the polysomnographic facility on the ground floor. And we also created short-term fellowship because that is to give opportunity to we envisage that so many other disciplines, people interested in sleep research should come forward and take the center forward. The comprehensive center of sleep had all these components envisaged: sleep disorder clinic, animal research, sleep specialty in neurology, psychology, pulmonology, ENT, and dental human research facility, circadian rhythm facility. And there in Trivandrum, you also have ISRO. We thought that human space program, they would require a lot of support. I mean, one of the main components they have to look into is the sleep of these spacemen. And somehow they had, this is the major plan which we had for the sleep center, which was approved by the DST, and the seed money was also given. Unfortunately, it didn't take forward because we did not get people who are prepared to leave all the other interests of theirs and totally dedicate their work on sleep research. And the only wing which succeeded was the animal research facility, which was headed by Kamla. And all these publications by my name may be there, but it is totally the effort of Kamla and her students. And in fact, one of the areas which she was concentrating on, effect of sleep deprivation during pregnancy. How does it affect the offspring? And the second area which she tried to do was the finding out the active principle of a normal Ayurvedic principle, that is alpha-acerone in Vaja, which is said to have sleep promoting influence. As you all know, even today we don't have a perfect medicine for insomnia. And the next effort was to have ultrasound vocalization to assess newborn stress, because many of these newborns born under a stressful mother also has some remnant of the effect of deprivation of sleep by the mother. Then, as I said earlier, that Kerala is one place peculiar in the sense it has the highest number of aging population. And being appointed as a representative of NAMS in Kerala, I thought I should do something on that direction. And we published, and by that time, COVID also came, which primarily affected the elderly population. And we had published several papers on this line of research, and I cannot go into details of it. In fact, I had a couple of papers which are highly cited. One of the paper was cited nearly 700 times. But anyway, that's when you don't want anything, you get everything you want. You were dreaming of one day. And so with this, I say thank you to all of you.
Thank you, sir, for that extensive and enlightening lecture. May I now request our Dean, Professor K.K. Verma, to present the award to Professor Mohan Kumar. Thank you, sir. As the oration draws to a close, it is an honor and privilege for me to extend a heartfelt vote of thanks for the conduct of the Kamla B.K. Anand oration. First and foremost, my deepest gratitude goes to our Dean, Professor K.K. Verma, for gracing the occasion with his valuable presence. Your time is precious, and we truly appreciate your generosity in sharing it with us. I also extend my heartfelt thanks to our orator and awardee, Professor V. Mohan Kumar, who is an inspiration to all of us. We are truly honored to have you with us again, sir. A special note of thanks to our esteemed former faculty and heads of departments who have joined us today, and faculty from other institutes in NCR and all over India who are here with us. It's an honor to share this occasion with all of you. I would also like to thank the registrar and academic section for arranging the logistics for today's function. Last but not the least, I would like to thank a big thank you to all the participants and attendees from Department of Physiology. And now we draw the function to a close. There is high tea, which is available in the dining hall adjoining the boardroom, where all of you are welcome to join us for high tea. Thank you.
Careful, careful.